Hello everyone! Today, we will be dealing with mutation, changes in genetic code. I am Ma'am Marian Arca Ilaw, your science teacher. What is mutation? Mutation is a change in the base sequence of DNA. It may affect only one gene or they may affect the whole chromosomes. It occurs when a DNA gene is damaged or changed in such a way as to alter the genetic message carried by that gene. Simply put, mutation is a change in the normal sequence of the DNA. How about mutagen? Mutagen is an agent of substance that can bring about a permanent alteration or permanent change to the physical composition of a DNA gene such that the genetic message is changed. Mutagen are substances that can cause mutations. There are two classes of mutation. Number one is point mutation. Point mutation happens through deletion, insertion, or substitution. Whereas chromosomal mutation happens through inversion, deletion, duplication, or translocation. Let's have this point mutation through deletion. In this process, one or more nucleotides is skipped during replication or otherwise exercise, often resulting in a frame shift. So here is the starting sequence or the normal DNA sequence, and then this part will be deleted. That is point mutation through deletion. Point mutation through insertion. In this process, one or more extra nucleotides are inserted into replicating DNA, often resulting in a frame shift. Insertion is the opposite of deletion because here the nucleotides or one or more extra nucleotides are being inserted during replication. Again, this is the starting sequence or the normal DNA sequence. And this part will be inserted in this starting sequence, resulting to this. So this is the inserted nucleotides. That is insertion point mutation. Point mutation through substitution. Substitution happens when one base is incorrectly added during replication and replaces or substituted the pair in the corresponding position on the complementary strand. It happens in missense mutation if a substitution changes the amino acid. Again, if a substitution changes the amino acid, it is called as missense mutation. If substitution does not change the amino acid, it is called as silent mutation. However, if substitution changes the amino acid to stop, it is a nonsense mutation. So here is the substitution point mutation. This is the no mutation with the DNA level and nitrogen bases of thymine cytosine, mRNA level of adenine guanine, and the protein level of lysine. The point mutations, specifically in silent substitution point mutation, the amino acid remains the same. The substitution does not change the amino acid, even if there are changes in the base pairing. Whereas in a nonsense point mutation, the substitution brings the amino acid to stop, even if there are changes in the base pairing in A adenine uracil pairing. And in missense mutation, the substitution changes the amino acids from lysine to arginine or threonine in this case, in this example. Okay, how about in chromosomal mutation through inversion? In this type of mutation, one region of a chromosome is flipped and reinserted. So this is the illustration showing the flipping and reinsertion of one region of a chromosome. From the normal set of chromosomes, this particular chromosomes or region of chromosome is flipped, nagkapalit. Chromosomal mutation through deletion. A region of a chromosome is lost, resulting in the absence of all the genes in that area. The normal set of chromosomes and then the deleted part of the chromosome, resulting to a mutated DNA. So this is the deleted part. Chromosomal mutation through duplication. This process shows wherein a region of a chromosome is repeated, inuulit, resulting in an increase in dosage from the genes in that region. This is the illustration of duplication. From the original chromosome, here, the blue color has been duplicated, resulting to a mutated chromosome. Chromosomal mutation through translocation. This process shows wherein a region from one chromosome is aberrantly attached to another chromosome, showing the normal set of chromosomes. Here, the process of translocations from this part, second part of the chromosome, the particular region is translocated to the fourth part of the chromosome. This is the activity number 1 about gene and chromosome mutations. You are going to match column A to column B. Write the letter of your answer on the space provided before the number. So you will write your answers on your answer sheet. In the second activity about mutations, you are going to complete the boxes below. Classify the type of mutation, whether deletion, insertion, or substitution, and as either frame shift, missense, silent, or nonsense. Hint: deletion or insertion will always be frame shift. This is the original DNA sequence. Now we are going to answer this number 1 DNA sequence, the mutated DNA sequence. These are the base pairing in the replication of the DNA in the mRNA sequence. Take a look at that the base pairing ATC and UAG changes from the original DNA sequence, which is ACC and UGG. And then the amino acid sequence here is methionine and then stop. This means that the DNA recognizes UAG as stop, which means that there will be no production of amino acid anymore. And what kind of mutation is this? It is a nonsense mutation. So for the rest of the activities, you are going to answer those, provided that the sequence number 1 is given to you as an example. Activity 3, sickle cell anemia. So anemia or sickle cell anemia is an inherited blood disorder where red blood cells or RBCs become sickle or crescent shaped. It causes frequent infections, swelling in the hands and legs, pain, severe tiredness, and delayed growth or puberty. You are going to analyze the DNA strands below to determine what amino acid is changed and what type of mutation occurred. From this normal hemoglobin DNA, we have CAC, GTG, GAC, TGA, GGA, CTC, CTC. So you are going to provide the normal hemoglobin mRNA or the DNA replication, and then the equivalent amino acid or protein. For the sickle cell hemoglobin DNA, there is now the mutation in the DNA sequence resulting to a sickle cell hemoglobin DNA. CAC, GTG, GAC rather, TGA, GGA, CAC, CTC. So you are going to analyze and provide the sickle cell hemoglobin mRNA. Look at this. The two sequence from the normal CTC, it becomes CAC. Therefore, the nitrogen or the nucleotide changes from CTC to CAC, resulting in a mutation. These are the references. Thank you and God bless us all.